Let's take a look at tyrosine kinase receptors now. On our overview slide, I told you that was our number 3 favorite. Let's go back to that real quick. Here's the overview slide, and as you can see right here, these are the tyrosine kinase receptors, and we labeled them as number 3. So here back again, our number 3 favorite for the purpose of this class, I should say. The tyrosine kinase receptors, and here's how they work. The signaling molecule right here, labeled as L for ligand, is binding to this receptor. This receptor has a single transmembrane domain, as you can see, this little spiral helix right here going through the phospholipid bilayer, and the other side of the molecule is a tyrosine kinase. Now, a kinase is an enzyme that phosphorylates other molecules. And so what this particular kinase will do, it's a tyrosine kinase. So tyrosine is one of the amino acids. And so it will take other proteins and phosphorylate, attach phosphates, as you can see right here, it will attach phosphates to the tyrosine amino acids of another protein. So right here, the phosphate comes from ATP, and the this tyrosine kinase will take a phosphate from ATP and slap it onto this protein right here, typically activating this protein. So here, I wrote this on the slide now. A kinase is absolutely a term that you have to know. A kinase is an enzyme that phosphorylates other proteins, typically other molecules in general. So it transfers phosphates. As you can see here, these tyrosine kinase receptors, once the ligand binds to them, it activates the kinase here, and then this kinase will phosphorylate other proteins. So then it leads to phosphorylated proteins. Phosphorylated proteins are typically activated proteins, and they go off and do other things inside of the cell. Kind of picture a kinase like a light switch. When you're turning on the light, so you're activating something. A kinase is kind of like a light switch. It turns stuff on. So now here, let's move on to our number 1 favorite, the G-protein coupled receptors. And go back to that overview slide, you will see I labeled this with a big number 1. This is your number 1 favorite. So the G-protein coupled receptors, GPCRs abbreviated, they're membrane spanning proteins. In fact, so they have 7 membrane spanning regions, 7 transmembrane domains is what it's usually called. And the cytoplasmic tail of the receptor is linked to a G protein. That's why they're called G protein coupled receptors. The G protein is a small protein. It has 3 parts. And here they call it a 3-part transducer molecule. So the 3 parts are different. That's why we label them as alpha, beta, gamma subunits. And or you can call it a heterotrimeric G protein. So this G protein is linked to the receptor. And once the ligand binds to that type of receptor, the G protein, usually a part of it, shoots off and activates whatever it's next to. And so when this G protein is activated, whatever is its next door neighbor that it's bumping into, it could open an ion channel, it could alter enzyme activity, it could do a number of things. This slide right here shows you what happens when a signaling molecule binds to a G protein coupled receptor. So here step 1 would be the signaling molecule binding to the receptor right here. This is the receptor portion. And so once the ligand binds to the receptor, then it changes the association to the G protein. The G protein will sort of be pushed away, and a portion of it shoots off and bumps into its next door neighbor. And then it really depends what that next door neighbor is, what's going to happen from here on out. So remember, we are dealing here with dominoes that start falling. And so let's say the next domino here is adenylate cyclase. It gets activated, and adenylate cyclase is an enzyme that takes ATP, takes 2 phosphates off of the ATP and makes it adenosine monophosphate from adenosine triphosphate. So this here is cyclic AMP. So it rips 2 phosphates off of the ATP, makes it AMP, and then it makes a cycle out of it, and then we call this thing cyclic AMP. Cyclic AMP is your most important second messenger. And cyclic AMP typically activates kinases. Here the example is given, it activates protein kinase A. And protein kinase A is a very important kinase. It activates all sorts of other kinases, and then you have the kinase activity and the kinase activity after that, and I'm not kidding, that's how it goes. These signal transduction cascades, they're basically activating kinase after kinase after kinase until you get some sort of a cellular response. And you can tell one little signaling molecule binding here leads to all of that, and that is huge. That's a huge output. So here you can go through the first step is the signaling molecule binds to the G protein coupled receptor, activating the G protein. The G protein bumps into adenylate cyclase, and the adenylate cyclase makes lots of cyclic AMP from the ATP, and then the cyclic AMP activates protein kinase A in this case, and then we get more kinases activated. Kinase upon kinase leads to a huge output. And because this is so important, we're going to do it in steps again. Here on this slide, you see only the signaling molecule binding to the G protein coupled receptor. Once that has happened, something happens to the receptor, and it causes the G protein, usually just a portion of it, to shoot off and to bump into its next door neighbor, which, in this case here, was the adenylate cyclase. The adenylate cyclase gets activated. Adenylate cyclase starts to grab onto ATP and makes cyclic AMP out of it. Cyclic AMP is your second messenger right here. Obviously the first messenger being this one right here. So here's now your second messenger. And then the second messenger will activate protein kinase A. And the protein kinase A as a kinase, as the name indicates, it's phosphorylates, all sorts of other things, turning them on, and it could very well be another kinase. So there would be a kinase activating by a kinase, and these things are literally called kinases, so that's kind of funny. But anyway, so then once you are all the way down, either at the nucleus or even in cytoplasm, you're activating all sorts of things, you get a huge output for a relatively small input, just that one signaling molecule, your first messenger binding to the G protein coupled receptor, and the result is all of this. Moving on to a variation of the theme. So here we have now a new next door neighbor to the G protein. Here, same starting point. Our signaling molecule binds to the G protein coupled receptor right here. It will cause a change to the receptor that causes a portion of the G protein to shoot off or dissociate and bump into its next door neighbor. Now the next door neighbor is called phospholipase C, PLC abbreviated. Now PLC is a lipase, and lipases, as you know, they digest fats or hydrolyze fats. Well, we are sitting here in the phospholipid bilayer, and phospholipid bilayers are made out of phospholipids. So a PLC, a phospholipase C, is a molecule that will take one of those phospholipids and chop its head off, basically. So the phospholipid, the head will come off, and it turns into a second messenger that's called IP3. That's this one right here. So that's the phosphohead group of a phospholipid. And the fatty acid tails of the phospholipid, they're then called diacylglycerol, which also are considered a second messenger. So the diacylglycerol a lot of times turns on protein kinase C, and the IP3 as a second messenger typically opens calcium gates and releases calcium from internal stores. Now calcium is also considered a second messenger that easily can lead to cellular responses. And the protein kinase C, well, it's another kinase. It will phosphorylate other proteins, which can also lead then to cellular responses. So this is a very popular system in cells that they use a lot of times, and it's a very powerful system. And because it is so awesome and so powerful, we're going to break it down in pieces and take a look step by step again. So here, starting point, our signaling molecule is binding to the G protein coupled receptor. This G protein coupled receptor gets then turned on. The G protein starts to shoot off, and it will bump into the next door neighbor, which in this case is PLC, phospholipase C. Phospholipase C gets turned on, and it's an enzyme that hydrolyzes phospholipids and splits them into the phosphohead group and the fatty acid tail, which in this case would be IP3 and diacylglycerol. So let's take a look at this here. So phospholipase C is splitting a phospholipid into diacylglycerol and IP3. This is the phosphohead group from that phospholipid. Now, both DAG, the diacylglycerol, and the the IP3 are second messengers. So DAG and IP3 are both second messengers, and as it turns out, the diacylglycerol will turn on protein kinase C, which then in turn phosphorylates other proteins. It's a kinase, so it will phosphorylate other stuff and lead to a cellular response by turning on kinases. And the IP3 is also a second messenger, and it will open calcium gates. Calcium will go down concentration gradients. It will be released from internal stores, and the calcium is also considered a messenger that then leads to cellular responses. So in conclusion here, the two G protein coupled receptor pathways that you need to know are the cyclic AMP second messenger pathway, and here the IP3 calcium pathway. We're not going to be talking more about the DAG, the diacylglycerol, but you do need to know about the IP3 as a second messenger and its ability to release calcium. So the most important G protein coupled receptor pathways are involving cyclic AMP, and then here the phospholipase 3 IP3 mechanism. Now moving on to a pathway that's not important for you guys, the integrants. I'm not going to say much about it, not to add to the confusion. So these the integrants are linked to the cytoskeleton. They also bind cells to one another, and for the purpose of this class we really don't need much about this. Okay, but here's something that you do need to know about, and that would be if a messenger molecule is binding to an ion channel, and that was our number 2 favorite from the original overview slide. So if the messenger binds to an ion channel, then this ion channel, the permeability of ions will be altered. It could either let more or fewer ions through, and that will alter the membrane potential of this membrane, and then you're going to have changes that are in the cell that are mediated through that. So the changes in the membrane permeability, this is what happens right here. The change in membrane permeability when you open ion channels, they will then lead to cellular responses ultimately. Here's an indirect mechanism that opens ion channels, and that will be a G protein coupled receptor that where the ligand, the messenger molecule, binds to the G protein coupled receptor, and now the G protein coupled receptor either directly or indirectly opens an ion channel. So the G protein could bump into an ion channel and open it up and alter the membrane permeability and change the membrane potential, or the G protein coupled receptor could activate some second messenger, activate a kinase, and then the kinase will open up the ion channel. So all of that is possible. So here in steps, we have the signaling molecule binding to an ion channel directly and opening this ion channel, or altering the permeability of this membrane, this channel to the to different ions. And then here our second possibility is that we have a the messenger binding to a G protein coupled receptor, and then the G protein would open or close an ion channel, altering the membrane permeability to a certain ion. And then here the third possibility is that the G protein coupled receptor activates some sort of intracellular kinase, typically some sort of a molecule, and that molecule could indirectly open up that ion channel. So 3 different ways you can alter membrane permeability, either directly over here, or indirectly with a G protein coupled receptor.